Bismillahirrahmanirrahim. Assalamu alaikum. Our today's topic is drug residues, veterinary medicine in relation to human health. In this lecture, I will be discussing with you, first of all, the background or preamble of this topic, and then which risk factors are involved for the development of residue in food producing animals, and they are listed here. Then the potential effects of veterinary drug residues on public health. And lastly, I will be discussing with you a residue avoidance, how we can avoid the residues in case of food producing animals. So let's start with the preamble. Human health is it's related directly to the environment, and in particular the nature and quality of the food. Quality of food from animal products, it is widely concerning public health agencies around the world, since veterinary drugs have played an important role in the field of animal husbandry and agro industry, and increasing occurrence of residues and resistance have become an interesting issues. So veterinary drugs or veterinary medicinal products are critically, they are needed to meet the challenges of providing adequate amounts of food for the growing world population. So as drugs improve the rate of weight gain, improve the feed efficiency, or prevent and treat diseases in food producing animals. However, the benefit of improved productivity from the use of veterinary medicinal products in food producing animals, it's not obtained without the risk associated with these medicinal products residues that remain in the tissues of treated animals at the time of slaughter, or residues in animal derived products, and it poses a health hazard to the customer. Antibacterial drugs and hormonal growth promoters are the main veterinary medicinal products that potentially contaminate foods of animal origin. So veterinary drug or veterinary medicinal products residues, it's one of the many global issues concerning food contamination. So rationally, there is no product coming from a treated animal should be consumed unless the entire drug administered has been eliminated. So this is called as a zero tolerance, where this concept is in fact equivalent to the idea of total absence of residual amounts. However, because of the improvement of analytical techniques, which meant that the value of zero became smaller and smaller, that depicts the limits corresponding to the sensitivities of parts per million, parts per billion, and parts per trillion. So as a result, by using the high efficacy analytical methods, for instance, using high performance liquid chromatography, HPLC, it can be concluded that there are nearly always detectable residues, but such residues are at an extremely low concentration and they are not inevitably toxic. So in this topic, I will be discussing with you the risk factors which are involved for the development of residue in food producing animals. So let's start with the first risk factor, which is the age of the animal. So weaning status and the age of the animal affect drug disposition. For instance, a study conducted on comparisons of the pharmacodynamics of norfloxacin nicotinate between weaning and unweaned calves revealed that the distribution of the drug, it did not differ between the two groups of calves, but the total body clearance time was increased in weaned calves, possibly due to increased weight from the presence of rumen fluid. So calves fed grain had shorter clearance times, approximately 4 days, for sulfamethoxine than unweaned calves. So the elimination half-life of tindazole is shorter in unweaned calves than in adult cows, while the elimination half-life of apramycin is longer in calves than in adult cattle, possibly due to immaturity of the drug clearance system. So I would say the age of the animal is very important for the elimination of these drug residues from the body system. The next factor is feeding. Diet can affect the bioavailability of drugs. For instance, uh, the effects of diet content on the bioavailability of orally administered fenbendazole to cattle and buffalo and fed dry hay. I mean, either with or without fresh green herbage. It depicts that animals which receive feed containing fresh herbage had lowered bioavailability of the drug. So fenbendazole stays in the rumen and is progressively released with digesta. And the presence of fresh herbage, it increases gut activity and the flow rate of digesta, which depletes the available stores of this fenbendazole in the rumen. So in regard to feeds, actual gut contents can also affect drug uptake and pharmacodynamics. The next risk factor, which is associated with the development of residues in food-borne or food-producing animals, is the disease status. So disease status of an animal, it can affect the pharmacokinetics of drugs administered, which can influence the potential for residues. This can occur either when the disease affects the metabolic system and consequently drug metabolism, or when the presence of infection or inflammation, it causes the drug to accumulate in affected tissues. So for example, cattle with acutely inflamed mastitis squatters, apramycin penetrates these areas of the body, and concentrations of the drug have been observed at 10 times over the level recorded from cows without mastitis. So in calves, with the proposed mechanism for these changes can be due to the liver function by fasciolosis, which change the processing of drugs through the liver. So now next factor is pharmacokinetics. The term pharmacokinetics, it refers to the movement of drugs into, through and out of the body, the time course of its absorption, bioavailability, distribution, metabolism, and excretion. So all these factors like absorption, which is the process, a compound in which a compound passes from its site of administration into the bloodstream. This is absorption. And in distribution is the process whereby a drug is transported to all the tissues and organs. So then comes metabolism or biotransformation, which is the principal mechanism of elimination for the transformation of drugs or xenobiotics into metabolites of the chemical reaction. And excretion, everybody knows, is the process by which the parent drug or its metabolites, they are removed from the body fluids. So pharmacokinetics at each level, it plays an important role for the removal or accumulation of these veterinary medicinal products. So another factor is improper withdrawal time. The withdrawal time, it's also known as the depletion or clearance period. It is the time for the residue of toxicological concern to reach a safe concentration, so as defined by the tolerance. Depending on the drug product, dosage form, and the route of administration, the withdrawal time, it may vary from a few hours to several days or weeks. So it is the interval necessary between the last administration to the animals of the drug under normal condition of used, and the time when treated animal can be slaughtered for the production of safe foodstuffs. Another important but last factor is incidence of veterinary drug residues. In many countries, these veterinary medicinal products, they may be used indiscriminately for the treatment of animal diseases, or they may be used as feed additives for domestic animals. Like in our country, these medicinal products, veterinary medicinal products are being used for weight gain or improving the growth rate of the birds and animals. So uh, 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 different studies, they have revealed that low level of heavy metals and gentian violet residues in other species like catfish, they have detected uh, uh, in slaughtered cattle, in the meat of slaughtered cattle have also been detected. Oxytetracycline and penicillin G from milk have also been detected. Um, uh, the ongoing threat of antibiotic contamination, it is one of the biggest challenges to public health that is faced by the human population worldwide. So such residues are spreading rapidly, irrespective of geographical, economical, or legal differences between the country. So let's discuss the potential effects of veterinary drug residues on public health. The first potential effect of these VMPs on public health is development of drug resistance. So human health can either affect through residues of drugs in food of animal origin, which may cause direct side effects, or indirectly through selection of antibiotic resistance determinants that may spread human pathogen. So resistant microorganism, they can get access to human either through direct contact or indirectly via milk, meat, or eggs. So as the bacteria of animal origin, they may either colonize human endogenous flora or superimpose an additional load to the reservoir of resistance genes, which are already present in man. So the potential for animal to human transfer of resistance, it existed. So clearly the use of antibiotic in livestock production or poultry production has been associated with the development of human antibiotic resistance. So the animals fed with the low prophylactic level of antibiotic, they may develop bacteria evolving resistance to this antibiotic during the preparation or consumption of food of animal origin. The next potential effect of veterinary drug residues on public health is drug hypersensitivity reactions. So it is defined as an immune mediated response to a drug agent in a sensitized patient, and drug allergy is restricted to a reaction mediated by immunoglobulin E. An allergic or a hypersensitive effect following administration of a drug, that is drug allergy, it is quite similar to that typified by allergic reaction to protein, carbohydrate, and lipid macromolecules. So allergic reactions to drugs may include anaphylaxis, serum sickness, cutaneous reaction, and a delayed hypersensitivity response to drugs. It appear to be more commonly associated with the antibiotics, especially of penicillin. Another potential effect of veterinary medicinal products on public health or human health is the carcinogenic effect. The term carcinogen refers to an effect produced by a substance having carcinogenic activity. Considerable confusion exists because carcinogen applies to substances that are so varied in their qualitative and quantitative characteristics. So the potential hazard of carcinogenic residues is related to their interaction or covalently binding to various intracellular components such as proteins, DNA, RNA, glycogen, phospholipids, and glutathiones. Another effect is mutagenic effects. The term mutagen is used to describe chemical or physical agents that can cause mutation in a DNA molecule or damage the genetic component of a cell or organisms. So several chemicals, including alkalizing agents and analogs of logos of DNA bases, they have been shown to elicit mutagenic activity. Um, Another effect is teratogenic effect. This term applies to drug or chemical agent that produces a toxic effect on the embryo or fetus during a critical phase of gestation. So these veterinary medicinal products, they have teratogenic effects, and different in different species these effects have been studied. For example, benzimidazole, a drug of oxfendazole, it has no doubt exhibited teratogenicity, but also exhibited a mutagenic effect. So another effect is disruption of normal intestinal flora. This is very common. The bacteria that usually live in the intestine, it acts as a barrier to prevent incoming pathogen from being established and causing diseases. So antibiotics may reduce the total number of these bacteria or selectively kill some more important species. The broad spectrum antimicrobials, they may adversely affect a wide range of intestinal flora and consequently cause gastrointestinal disturbance. For example, the use of drugs like flunixin, streptomycin, and tylosine in animals, and also the use of vancomycin, nitroimidazole, and metronidazole in humans, they are known for this kind of effects. Now, the question is, after knowing the risk factors associated with the development of residue in food producing animals, and after knowing the potential effects of these veterinary medicinal products on public health, the question is how we can avoid this these residues effects, or I would say residue avoidance, how we can attain. The first principle is based on pharmacological basis, is that we must especially those personas which are involved in farming, whether they are rearing the large animals, whether they are rearing the poultry flocks, all must know or consider the half-life of a medicine or an antibiotic. So it is the time it takes to remove 50% of the drug from the animal and is used to estimate the withdrawal time. So a drug with a large volume of distribution, it generally has relatively good tissue distribution compared with a drug of restricted distribution. I would say that the half-life of a product, veterinary medicinal product, must be kept in mind while adding it in the animal diet, no doubt as a prophylactic measure, or whether we are at a metaphylactic level. So control and preventive measures are also, for example, in general, the residue control strategy, it is based on a two-step approach. The first is the detection of residues using sensitive tests with a low rate of false negatives. And then confirmation, which requires quantification against these products with a low rate of false positive. So the residue prevention strategy is based on preventing the entry of violative residues in meat or milk intended for human consumption by a proper drug use guide, which has been developed for use by both veterinarians and food animal producers. So, in which hard health management, use of only approved drugs, establishment of valid veterinarian client patient relationship, proper drug administration and identification of treated animals, proper maintenance of treatment records and identification of treated animals, having proper drug residue testing capabilities, and creating awareness of proper drug use and methods to avoid marketing adulterated products, principally educational. So all by using all these strategies, we can avoid the antibacterial residue presence in the different food products in different food producing animals. If I want to, if I conclude or recommend the use of veterinary drugs in food producing animals, it has no doubt a potential to generate residues in animal derived products, and it definitely poses a health hazard to the consumer. So, I mean, we can see that these antibacterial or veterinary medicinal products are being used, no doubt, for obtaining the growth rate of the animals. But also, we use these medicines for treating the animals against different diseases. So the conclusion is that we should limit the use of these antibiotics and find some alternative resources. For example, if we want to get a very good growth promotion in animals, we not only we have the option of antibiotic growth promoters, but we can also use organic acids. We have some probiotics, prebiotic, symbiotic products, and we have phytobiotic products. So these are some conclusions and recommendations regarding the veterinary residues, especially if we consider the human health from the perspective of food producing animals. So that was all. Thank you very much. Allah Hafiz.